Good morning, everybody. This week we are redoing those fluorescence transfections. So I want to have 3 technical replicates, as in 3 different times that I did it totally independently to make sure the efficiencies are the same each time. Not something that I'm doing by mistake to one tube or another. So that involves some gene editing, it involves some tissue culture, and then on a second note, we're going ahead with the molecular cloning to continue cloning in new pieces into those same plasmids. Wish me luck. Good afternoon, everybody. So today was just a quick little day. Basically, I said this morning that I want to do my transfections 2 more times. So my gene editing 2 more times to account for any mistakes I may have made. Maybe one lot of cells got slightly warmer and did slightly better, or just a fluke of the way things were. So I want to do them 2 more times. Ideally, I would have started them today, but my cells weren't grown up enough. I didn't have enough of them. I would say they're about 40% confluent, so I'll check them out again tomorrow, but I suspect they're not going to be fully confluent until Wednesday. What I did get done though was sending my sequencing off, and I also did a touchdown PCR for the first time, which is really clever. I'd vaguely heard about them before, but basically a touchdown PCR starts at higher annealing temperatures and works its way down to preferentially amplify the most similar sequences. I haven't really explained PCR on this channel before, and I'd rather wait until I have actual footage of me doing it before I put it up on the channel. Basically, you're cycling through different temperatures that allow your primer and DNA to stick together, then elongation to occur along the original DNA, and then separation, and having that happen over and over again. So starting at a higher temperature for the annealing, which is the sticking together of the primer and DNA, is meant to reduce non-specific binding between the primer and DNA. So anyway, I have reached the noisy road now, so I'll leave it there for today. See you tomorrow. Good morning, everybody. Today I have to do a gel electrophoresis of my PCR product from yesterday. If that's all looking good, I'm sending it off for sequencing. And I also have to check out my cells to see if they're ready for editing, but I don't think they will be. So I've got a bit to do, but there's also a big meeting at my institute at 11 o'clock, and of course all meetings are via Zoom right now. So I've got to be done in time to be home by 11 because I don't have a computer here to watch the meeting. I mean, if I don't make it in, the worst that happens is I have to come back in afterwards to get some experimental work done. So it's not the end of the world. But I've got to say, once I get home, I don't exactly want to come back out into the cold. And there's a few papers I'd like to read this afternoon, 2 specifically, so we'll see if we can get out quick this morning. Hello everybody. Okay, so mixed results this session, but I did get some filming in, so you'll actually get to watch those results happen. So today I ran my PCR product from yesterday on a gel to make sure the pieces were the right size, and the goal was then to send them for sequencing, which I did do. But one of them came up empty, which is a bit weird. But even weirder, my no template control from yesterday, so a PCR with no DNA in it, came up on the gel at the right size. So it's very tempting to think that maybe I just mislabeled the tubes. One of them's gRNA4 and one of them's my no template control, but I can't just assume that. So for the moment, I've sent off for sequencing for the ones that I'm certain of. But I'm a little bit confused about what's happened with gRNA4. Either I've stuffed up the extraction or I've stuffed up the PCR, but other than just straight up not pipetting in, I'm not really sure. I don't have many ideas as to what's happened. Anyway, I am at the Loud Road, so I'll see you tomorrow. Good morning, everybody. I'm starting quite a bit later than normal today. It's nearly 9 o'clock because I couldn't get time in the tissue cabinet before that. So today I'm doing that second round of transfection, so second round of gene editing to see if we get similar results. It's still only the second time I've ever done this, so I'm gonna take it quite slow, and I've got my supervisor nearby so that I can ask him questions if I need to, but I'm feeling good about it. I'm gonna grab some footage today so I can show you at least a little bit of what it looks like to edit cells. So I guess I'll see you in there. Good afternoon, everybody. So 3 things happened today. Well, that's not fair. 1 thing happened today 3 times, and I am upset about it. It's like week 4 of my PhD, I don't even know. And I remember before this all started saying, you know, everyone gets too hard on themselves in their PhD. Everyone takes things so personally, and don't do that. You know that happens, so don't do that. But here I am. Oh boy, how do I explain? So basically, when you're doing CRISPR, there is a stage where you kind of electrocute your cells with the guide RNA. But if it goes wrong, if there's a bubble of air in it, the electricity can go through those bubbles of air and arc. So basically fry your cells. And out of 12 attempts, 3 of mine arced, including one that arced previously and didn't do so well after that. And we're not sure if it's because it arced or because of anything else, like because the guide RNA isn't as good or whatever. So the one that I really wanted to know what it would react like if it didn't arc, and it's not anything that I'm aware I could have done differently. Like if there are big obvious bubbles in it, the machine just refuses to shock it. It can tell when I watch them more carefully, so I'm just not sure. Everyone just seems to say, you get used to it, there's a way to it, you pick up the skills, but I don't have those skills yet, so I'm frustrated. But anyway, I did film the whole thing, so at least I can show you what it looks like when the machine arcs. But I wish I couldn't. So anyway, this isn't like the end of that or anything. I've still plated them out and they'll go ahead and we'll see what they look like tomorrow and next week and all of that. Just every little extra factor you have to consider makes analysis more complicated. It just makes for a less clean story. And I think the cleaner your story is in the PhD, the easier it is at the end. And this is just the beginning. So hopefully I get so good at this and it's barely any effort anymore. That's probably not likely to happen. So in conclusion, today feels like you can feel like you're doing everything right and it can still go wrong, which is unfortunate, but I'm gonna go home and have some lunch and that'll probably make me feel better. Okay, so I don't know if I said yesterday, but I made up a plate and today I'm making up an overnight culture from that plate. What else am I doing? What else am I doing? I know I don't have much to do today. Oh, the other thing I have to do today is refresh the media on the transfections I did yesterday. So they've had time to kind of get settled down on the bottom of the plate. So I'll take off the old media and I'll put in fresh media that's got new nutrients for them, all of that. So that will take like some time, but isn't very complicated. It'll be a good chance to check out how they're going, especially the ones that I fried a little bit. So yeah, mostly just drudge work today. Good afternoon, everybody. Today was pretty simple. I did get colonies on my plate, which is fantastic. So they are now in for an overnight culture, and tomorrow I'll do a Maxiprep. I haven't done a Maxiprep before, but it really just sounds like a really big mini prep. Not too worried about that. And also my transfected cells are looking pretty good. They weren't ready to be split yet. I just refreshed their media, so they've got some nutrients, but they're looking really healthy. They'll be ready for splitting tomorrow. Pretty uneventful day, so I'm gonna go home, have some lunch, and read some papers. Good morning, everybody. So it is 8 a.m. I'm ducking in for the first time this morning. I'm going to pellet down my overnight culture from yesterday. Then I'm going home for our lab meeting, and then I'll be coming back in later this afternoon to do a Maxiprep for the first time. I'll see you in a bit. Good morning again, everybody. My overnight cultures had grown fine. I spun them down fine, even in the big scary machine. And now I'm heading back to have some brekkie and go to lab meeting. I don't really have much to show for my work this week. It's all been about doing the experiments and I don't really have the results yet. So today, lab meeting is going to be about listening and absorbing. I'm still very much getting used to what everyone else's project is, trying to place them all on the different projects that are happening in my lab. Good morning again, everybody. Big change of plans. My supervisor's Wi-Fi is down for at least 24 hours, which is no good at all, but it does mean that we don't have our lab meeting. So instead of having to be in till like 4:30, 5 p.m. tonight, I'm now rushing in to try and get to a 10:30 tissue culture booking. I think it's about 20 past 10 right now. So I guess I will tell you about what I'm about to do when I finish it. Good afternoon, everybody. So today was a really productive day. I did a Maxiprep for the first time just then. So a Maxiprep is getting more plasmid out of more bacteria than a mini prep. And it was actually really funny because, you know, for a mini prep, your tube sizes are like this much. The Maxiprep tubes are like this big because you're just going to use so much more, which makes sense. But it was just very funny to see. And the kit was like quite large. I think it went really well. I had a visible pellet at the bottom after the centrifugation steps. Seems like I managed to get my plasmid out. It is the weekend and it is time for me to not think about my science too much. So do subscribe and comment if you are interested in seeing more of this content. And other than that, I will see you guys next week.